Okay, welcome you all. Today we are going to discuss about little bit about host cells. Then we'll move on to the next topic, that is methods of gene transfer. And third, if time permits, we will discuss selection of recombinants. Okay. So before going to the topic, we'll just recall our previous uh, topics so far that we have been discussed, such as we had discussion on the basic steps of RDT, and we also discussed the different tools used like transfection enzymes, other enzymes, vectors, and today the most important technique is topic is host cells. Host cell. Host cell means the cell to which you are going to transfer the recombinant vector. हमने recombinant vector बनाया और अभी वक्त आ गया उस recombinant vector को हमें एक host cell में भेजना है. Now the question comes, what type of host cells would we select, and what are the cells which can be used as host cells? Okay, there are different bacteria and there are different animal cells and plant cells which can be used as host cells, but among which हम किसको लें? और अगर किसी और सेल एक सेल को आप सेलेक्ट कर रहे हैं तो why would you select that? Okay, so these are the things which we are going to discuss now. The first thing is, for a prokaryotic gene, for a prokaryotic gene, we always select a prokaryotic host. The host will also be prokaryote. The reason is, every type of gene, for example any prokaryotic gene, it is having a special structure. In the last year we had discussion on the structure of gene. There we studied that prokaryotic genes are different from eukaryotic genes. So prokaryotic genes are best suitable in prokaryotic host. So one of the very commonly or very popular host is E. coli, Escherichia coli. Why Escherichia coli? सबसे इम्पोर्टेंट उसका फ़ायदा क्या है और कैरेक्टरिस्टिक क्या है? It divides in 20 minutes. Majority of bacteria, they divide within 20 minutes. So this is one of the major advantages of selecting E. coli as a host cell. And moreover, there are some characteristics which makes E. coli has the best suitable host. One is its genetic makeup, its genome, genetic makeup is extensively studied. Extensively in the sense they are studied well. Their genome we know very well. E. coli genome, each and every part we know very well. So it is easy to transfer gene onto that or into that cell, as well as we can find out where our cell gene goes and all. Their genetic makeup is extensively studied. Second important characteristic is it is easy to handle. Easy to handle in the sense easy to grow, easy to make further generations. Okay, easy to grow in any of the conditions. Third important characteristics is they accept a range of vectors. These E. coli accept a variety of vectors. So these are some of the characteristics made E. coli as a favorable host for prokaryotic genes. And another important thing is for eukaryotes, prokaryotes and eukaryotes, they have different types of genes. For eukaryotic gene, the most extensively used host cell is yeast. जैसे हमने प्रोकैरियोट के केस में ई कॉली में और ई कॉली के डिफरेंट कैरेक्टरिस्टिक्स बताया, वो सारे कैरेक्टरिस्टिक्स ईस्ट में भी एप्लीकेबल है. Easy to handle, easy to grow, accept variety of vectors. This is why we select yeast as a favorable host for majority of eukaryotic gene transfer experiments. And moreover, eukaryotic genes are having a lot of additional mechanisms like post-transcriptional modification and all, and post-translation modification, and yeast is one of the simplest eukaryote. All these things makes yeast as one of the best, favorite hosts in all the recombinant DNA experiments. Okay. Now, apart from these, you can use any cell as a host, any animal cell or any plant cell as a host. ज़रूरी नहीं है कि आपको हमेशा yeast and E. coli यूज़ करना है. You can use any cell as a host. You can transfer gene into any of the cells. But one among them, most popular is for prokaryote it is E. coli, and for eukaryote it is yeast. Okay. Now this is regarding host cell. Now we will move on to the next topic. Next topic is regarding gene transfer or introduction of rDNA into the host. Now we discuss the methods of introduction of recombinant DNA to host. So there are so many methods. Among them we will discuss five major techniques. The first one is transformation. This is one of the important techniques. और कभी कभी हम recombination को भी transformation बोलते हैं. Okay. The transformation is such a common method, and how this is being done? Normally a bacterial cell would take up a plasmid from the surrounding without any special hesitation, or we can say without any special treatment. This is generally. But our favorable host E. coli will not take it, and this E. coli requires some additional techniques, okay, or additional treatments. So transformation, let us discuss transformation first. It is the method of gene transfer where naked DNA is directly taken up by bacterial cells. But for E. coli, they won't take it easily, or they won't take naked DNA so easily. And for E. coli, there is a special technique or a special treatment developed by Mandel and Higa. They said that this E. coli is to be made competent by treating them with cold calcium chloride. You have to treat E. coli with cold calcium chloride. Then they become competent. So what is the word meaning competent is? They are ready to accept foreign DNA. So according to Mendel and Higa, they have designed a technique that E. coli is treated with cold calcium chloride and made them competent. Just after cold calcium chloride, you give a heat shock, and then accordingly you will be giving mixing the recombinant DNA along with the host cell, then host cell takes up. This is only for E. coli. Okay, so this you need to keep in your mind. Now, next technique is transfection. Second technique is transfection. So here, this foreign DNA is mixed with some charged substances. Our DNA is mixed with some charged substances like calcium phosphate. Another thing is liposome or DEAA dextran. These are some charged substances. You mix your DNA with this, and then the cells will take up it. Generally, transfection is the word used for plant, animal, and other cells. For bacterial cells, normally we say transformation. But if you use another host like plant cell or animal cell, into which you are giving naked DNA along with some charged substances, and that cell takes up it, and this method is known as transfection. Whereas if you do the experiment with E. coli, if E. coli is taking DNA from the environment, then it is termed as transformation. So see the difference between transfection and transformation. Moving on to the next technique. Next is electroporation. 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 As the name says, it is involving electricity. The cells are kept in a chamber, and to which we pass electric shock. Suppose these are the cells, and we give them electric shock. When we give electric shock, the cells will generate some transient pores. Transient pores in the sense they develop some pores which are temporary. See the term transient pores. Cells form some transient pores. Now if you mix your rDNA into this medium, and these rDNA can go inside the host cell. So electroporation is a method of gene transfer where cells are kept in a chamber, and given a mild electric shock. So when we are giving mild electric shock, there are formation of transient pores through which your rDNA can enter. Okay, moving on to the next, that is micro injection. Next, micro injection. Micro injection. This is another method of gene delivery into the cells where we directly inject the the DNA into the host cell by using a needle. So this is injecting needle or micro syringe. To hold the cell, we use another pipette that is known as holding pipette. With a holding pipette, we hold the cell and to which we are introducing our gene of interest directly into the nucleus. सेल के अंदर न्यूक्लियस है, हमारा rDNA directly न्यूक्लियस के अंदर भेज देते हैं. So this is micro injection, and please make a note. This has to be done with some experts who can easily handle the cells under microscope. Now moving on to the next technique, that is biolistic. Next technique है biolistic. This is also known as gene gun, which is also known as particle gun. In this method, the cells are coated with, or recombinant DNA are actually coated over tungsten or gold nanoparticles. हमारा rDNA इसके ऊपर कोट करेंगे nanoparticle, and they are loaded on a gun. Gun in the sense this is a particle gun. They are loaded into this, and these are shot against the cell. This is literally just like shooting, but with a mild force, so that these particles would enter the host cells nucleus तक जाएंगे वो, and since these particles are of tungsten and gold, they are very less reactive. They don't react with the cellular contents, but these DNA can be easily delivered into the nucleus. This is biolistic or gene gun. So apart from this, there is a bacterium which is also known as natural genetic engineer. This bacterium is Agrobacterium This Agrobacterium is tumefaciens. This Agrobacterium is also known as natural genetic engineer. Because this bacteria is having a special plasmid. This plasmid is known as TI plasmid, which induce tumor in plant cells. So this TI plasmid is having a small region known as tDNA. इसके एक छोटा सा पोर्शन tDNA कहते हैं. ये DNA यहाँ से प्लांट के अंदर भेज देते हैं this bacteria, only tDNA, and causing the disease tumor. This is tumor inducing plasmid. So this plasmid can also be used as a vector, or it is very commonly used for plants. So these are the major methods of gene delivery. Once again we'll see. First one is transformation, which is normally told for, or normally this is used for bacterial cells. Mendel and his आगे technique याद रखना. The second one is transfection, mixing with some other charged substances. This is generally done for eukaryotic cells. Electroporation is a method where a mild electric shock is given and then the rDNA is transferred. Microinjection is using a syringe. Bialistic is another method where the particles are bombarded. And the last one is agrobacterium mediated gene transfer. So these are the topics. I hope you got it clear. Thank you.